Hi everybody. Today we're going to be talking about DNA and structure, so looking at the actual physical structure of the DNA molecule, the shape of it, the actual components of it, and just once again the physical shape, and also comparing it to RNA and RNA shape and structure. One thing that we should already remember from the first unit is that DNA and RNA are both nucleic acids. So this is the very important macromolecule that has a role in storing and carrying genetic information. And that's what this entire unit is about, is how does DNA and RNA, or what are the functions of DNA, RNA, and how does an organism use this information to actually create proteins and survive in the environment? As we should already know, the shape of a DNA molecule is this double helix shape, this twisted ladder shape. And it consists of these two coils, so there's two strands of DNA that are twisted around each other, creating this double helix molecule. The actual structure of DNA is made up of nucleotides, as we already should know. The monomer, so they are the monomer unit of DNA, so nucleotides are the monomers of DNA. We also already mentioned this back in the first unit, but as a refresher, each nucleotide is three components. You have a phosphate group, which is a PO4 group, so one phosphorus molecule and 4 oxygen molecules. You have a nitrogenous base or a nitrogen base, and then you have this 5-carbon sugar. So a phosphate group, a nitrogen base, and a sugar, this pentose sugar. And each one of these little nucleotides is what you link together in a big long chain to form that nucleic acid, specifically that DNA molecule or RNA molecule. And it's these nitrogen bases that are actually all different. There's 5 different nitrogen bases. 4 of them are found in DNA, 4 of them are found in RNA, and there's one that's different between the two of them. If we look at the actual molecules, the actual shape of DNA, so if we were to untwist that ladder and have this not twisted shape, we can see that the outside edges, the backbone as we call it, is made of alternating phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. These molecules, these phosphate sugar molecules, are held together by covalent bonds, which are extremely, extremely strong. So this backbone, that's why it's called a backbone, is not going to be able to be ripped apart. This is going to be a structure that's going to stay together because it is covalently bonded together. The inside rungs of the DNA molecule consist of the nitrogen bases. And they are held together by hydrogen bonds, which are much weaker. And we actually can separate these two strands by breaking these hydrogen bonds, just like with water molecules. So the backbone is phosphate, sugar, phosphate, sugar, phosphate, sugar, when covalently bonded together, while the inside rungs of that ladder are the nitrogen bases that are only hydrogen bound together, so they're much, much weaker. There are 4 different bases inside of DNA. You have adenine, you have guanine, you have cytosine, and you have thymine, or we usually abbreviate A, G, C, and T. And you can see the shapes here. So these nitrogenous bases, these nitrogen bases, the adenine and guanine actually have 2 rings, a hexagon and a pentagon, while the cytosine and thymine only have 1 ring, which is a hexagon. And the technical name, adenine and guanine, are what we call purines, and cytosine and thymine are what we call pyrimidines, which you really don't need to know currently. However, in ACE biology, that is, you are expected to know that adenine and guanine are purines and have this double ring structure, while cytosine and thymine are pyrimidines and have this single ring structure. So these guys are what's actually going to form the rungs of that DNA ladder, the inside part of that DNA ladder. And these nitrogen bases, this actually creates the code that makes UU. So all DNA molecules have the phosphate and sugar backbone, but it's the order of these nitrogen bases that actually make UU. That is what your DNA is. If we were to once again look at the entire structure, DNA is antiparallel, which means they actually run in opposite order. So the way we describe this is one strand runs 5' to 3', and therefore the opposite strand, the complementary strand, the other strand of the DNA molecule, runs 5' to 3' in the opposite direction. So this end is 5', this end is 3', this end is 5', this end is 3'. So it's almost like a road where they're going opposite directions. The easy way to remember which end is which is the phosphate end, always is the 5' end. Phosphate 5, they both have an F sound, so that's how you can remember which end is which. And then the OH group on the end of the sugar molecule is the 3' end. But honestly, if you just remember which one's the 5', well, the other side will be the 3'. And the actual 5', 3' numbering deals with chemistry and deals with how many carbons there are in the sugar. So we actually can number the carbons in this sugar molecule. This is a pentose molecule, so there's 5 carbons. Each little point is a carbon. So if we look here, we have 1, 2, 3, 4, 5. I'm not going over why it's ordered in that way, but we can see that the phosphate group is attached to the 5th sugar, while the hydroxyl group is attached to the 3rd carbon molecule. And that's where they get the numbering, but you don't need to understand that. I just want you to understand this idea of antiparallel and the phosphate group being 5' and the hydroxyl group being 3', and that they run in opposite directions. Watson and Crick was the famous scientist pair that discovered the shape of DNA molecules, so they're the ones that decided and had evidence to back up that this molecule was an actual double helix shape. And they said that the strands are complementary to each other, meaning that there's actually a pairing up that occurs, that you can't just have an adenine pairing to an adenine, that there's an actual complementary pair that orders the DNA and keeps it together. So the nucleotides on the template order themselves based on what we call Chargaff's rules. And Chargaff was another scientist who came up with this. When he was looking at ratios of these 4 nitrogen bases, he discovered that there were an equal amount of certain ones, and he was the one that came up with these base pairing rules, which is adenine will only pair to thymine inside of DNA, and cytosine will only pair to guanine inside of DNA. So if we were to practice that, if we have this one strand here, which is the template strand, so this is just one strand of the DNA molecule. So we have adenine, adenine, thymine, cytosine, guanine, cytosine, thymine, adenine, cytosine. Based on one strand of DNA, you can tell me what the other strand is because they have to follow this base pairing rule. So if you have a adenine here on the other strand, which would be the 3' because they're opposite, you would have to need a thymine because A goes to T, adenine to thymine, thymine to adenine, cytosine to guanine, guanine to cytosine, cytosine to guanine, thymine to adenine, adenine to thymine, thymine to adenine, adenine to thymine, and then finally cytosine to guanine, and then that's your 5' end. So you do need to know the base pairing rules. You need to go A goes to T, and C will only go to G in this base pairing. It's these hydrogen bonds that are going to keep this DNA molecule together and keep these strands together. So once again, these nitrogen bases are what's forming the inside part of the DNA molecule. And then you have your phosphate sugar backbone that's on the outside part of it. And a quick review or quick comparison to RNA structure. The difference, this is mostly the differences between the two of them. So DNA has deoxyribose as its sugar, while RNA has a different sugar. It's just ribose. That's why deoxyribose DNA has a D in it, while RNA is an R because it has ribose sugar in it. So RNA is ribonucleic acid compared to deoxyribonucleic acid. So when you look at its nucleotide components, it has a ribose sugar instead of a deoxyribose. It still has the phosphate group, and it still has the nitrogen bases. However, the base pairing rules are slightly different. RNA and DNA both have adenine, they both have cytosine, and they both have guanine. However, DNA has thymine, the T. RNA has uracil, U. So the base pairing rules are adenine actually pairs to uracil in RNA, and cytosine pairs to guanine. DNA is double-stranded, so you have those two complementary strands. RNA is actually single-stranded. And to quickly recap that, between the DNA and RNA, DNA is double-stranded, RNA is single-stranded. DNA has deoxyribose as that sugar inside of its nucleotides, which is why it's DNA. RNA has ribose sugar, which is why it's RNA. And for the base pairings, in DNA, adenine pairs with thymine, A to T. In RNA, adenine pairs with uracil A to U.